Hi everyone, welcome back. In the last video, I talked a little bit about the basics of collecting a sample for RNA sequencing, and then what does the file actually look like when you get your results of your RNA sequencing. So we're just going to jump right back in to kind of quality control of that file, which, just as a brief refresher, this is a FASTQ file. That is what you're going to get as a result of your RNA sequencing. It has 4 different kind of lines for every single read. It'll have an identifier right at the top that uniquely identifies that read. It'll have a nucleic acid sequence, which tends to be of a pretty standardized length for your samples. So, for example, for mine, it was around 75 base pairs. You're definitely aiming for over 100 base pairs on average, though. You'll kind of have this separating line, and then you'll have your Phred score, which is the quality for each of your bases along this read. So once you have that file, the first thing you need to do is check the quality. So the Phred score is really not that intelligible on itself. It's kind of just a random mix of letters and numbers that indicates the quality based off of some key that's being used by the sequencing company, but it's not very easy to just look at and assess on your own. So normally a program called FASTQC can be used to check the quality of your data. Now, in my experience, actually, when you receive your FASTQ file, you'll also receive an accompanying FASTQC file for each of those files. So they'll often do this already in advance, the sequencing company, and send that over to you as well, which is nice because it's just one less program to have to run. So I'm not going to talk in detail about all of the graphs that are involved in FASTQC. I can make another video where I have the graphs and examples and we talk through what all of them mean if people are interested. But I'm just going to talk about some kind of main graphs that you might want to look at if you're doing RNAseq analysis. So the first would be the per base sequence quality. So just to briefly give you an idea of what this graph would look like, and I'll also link to a resource that has a really nice walkthrough of all of these graphs. But on the x-axis, you'll have the base position. So as I mentioned before, all of your reads are about the same length. So this will just indicate what base pair in that read are we looking at, from 1 to however long your base sequence, your reads were. For example, mine, the longest was 76. So this would be from 1 to 76 for me. So for example, in this sequence, this would be base 1, 2, 3, 4, 5, and so on, right? So just what base are we looking at? And on the on the y-axis, there's a quality score, or the Phred score. And often these graphs will actually have lines that delineate kind of a good quality Phred score, a medium quality Phred score, and a bad quality Phred score. And really what you're looking at on this graph, I can just draw kind of some sample data of what it might look like. Maybe you have really good quality at the start, and then it kind of drops off at the end. So generally, this would be good. A higher Phred score is good, and a lower is bad, right? So maybe my data looks like this purple line. I have good quality bases, good confidence in my base pairs until, maybe I drop off around 60 or something like that. And this is pretty common to see the quality drop off at the end. And in my detailed FASTQC video, I can get into why that is. But you want to make sure that you don't have a large number of bad quality bases, because again, this quality indicates how confident we are that that is the correct base. So you could have kind of maybe the last, for example, what I'm showing right here, the last 16 bases in all of my reads are poor quality. I don't necessarily want to include those in my future analysis because there might be some confusion of what the correct bases are, and it might kind of start to overshadow my good data and kind of confuse some of the later programs on what the true bases are in those segments. So we might want to check out this graph and potentially proceed with trimming off kind of these last 16 base pairs from all of our data if we consistently see that they are poor quality. So the other two graphs that I would focus on for RNA sequencing data is going to be the adapter content and also the overrepresented sequences. A lot of the FASTQC graphs are a little bit more catered to analyzing analysis of DNA transcripts rather than RNA transcripts. So they're not going to deal very well with kind of the replication of sequences, right? Because you're looking at an RNA transcript, there could be many copies of a single gene that are being transcribed. That's not problematic, but FASTQC could flag that as problematic. And there's also many other graphs that I'll talk about in my FASTQC video that I'll explain kind of why they might not be the best thing to look at for RNA-seq. But for now, we'll just focus on what is good to look at, which again is going to be the adapter content, the overrepresented sequences, as well as this per base sequence quality. So the adapter content is going to tell you whether or not there are any sequencing adapters detected. So when your RNA samples are sequenced, they're going to add on adapters, which are short sequences that are going to help with that sequencing process. Again, I can talk more about this in an RNA-seq video, but they'll add on these adapters, and sometimes they can be left on and included in the sequence that's actually measured. So maybe the last, 10 to 15 base pairs of your sequence is actually not the RNA that you collected, it's these adapters that are present on the ends of your sequences. So that's not in the actual endogenous DNA or RNA of the animal. So we don't want to include that as we go on to analyze this data. So FASTQC can actually detect whether or not these adapters are present. And you'll see that there'll be kind of a graph that indicates the presence of those adapters. So if you see that there are adapters present in your sample, you're going to want to go on to trim those adapters off, because that is a big priority, is making sure that the adapters are gone. And then lastly, we have overrepresented sequences. So this is a bit of a messy one with RNA sequencing, because this essentially just is going to flag whether or not you have some sequences appearing a lot more frequently than others. More frequently. Again, with RNA, because these are transcripts that could have been transcribed like a varying number of times in the cell, naturally you'll have more of certain genes than others being transcribed. So this can be a little bit deceiving. This can also be problematic with poly A tails. So, for example, in my data, I had a bunch of overrepresented sequences that were flagged. and I didn't really know why or what was going on. I later realized that every single one of those sequences had maybe 20 to 30 base pairs that were just T's or just A's right at the end. So that was actually just a poly A tail showing up at the end. And because it made up such a significant portion of my read length, it was like 30 bases out of 75, it was being flagged as like a repetitive sequence in my data, even though it was just a poly A tail. So that's something to look out for. I've read pretty extensively about whether or not these poly A tails need to be trimmed, and it seems like the consensus is that they do not need to be. So you don't need to necessarily trim for poly A tails, but there may be overrepresented sequences that are not due to poly A tails and are not due to primers. So the best thing that you can do in that case is a process called BLAST. And I usually use BLAST N for this, N standing for nucleotide. I'll include the links to kind of all of the websites where you can do these things. BLAST has a very nice graphical user interface where you can just upload your sequence and you can choose a reference genome. BLAST is really just going to compare your sequence. So in this case, it would be whatever overrepresented sequence you see that you're going to copy and paste as kind of, it's called your query, because it's what you're searching for. And it's going to compare that to reference sequences. So this could be from specific genomes. If you have organisms that are similar to what you study, for example, if you study mice, then you could blast it against a mice or a mouse genome, and that would be really helpful. And this can kind of help to identify the sequence. So it tells you whether or not there are any close matches in terms of the actual base pairs in the sequence, and what that sequence might be. Sometimes it can be contaminants, which is why it's always good to blast your sequences. For me, I had one overrepresented sequence that I performed blast on. Again, I'll link kind of the where you can find the website where you can perform blast. And I compared it against a reference genome for my organism that I was using, and I found that it was just a really like common insect gene that was actually popping up in many different insects very repetitively. So just a highly conserved gene that's transcribed a lot, so not a big problem. But you might have cases where you have contaminants in your sample or something similar where you blast that sequence and you're not finding it, and then that might be a time to kind of investigate further. Now, when I say investigate further, I didn't have to, so I don't necessarily know what that would look like, but I'll link some resources that might help you figure out how to proceed from there if you were to find, for example, a contaminant. And a lot of that might go into trimming, which is the next video that I'm going to make. So thank you so much for watching so far. In the next video, again, I'll circle back to, now that we've checked the quality of our data, we proceed with trimming off what is, either not high quality or isn't serving us, like adapters.